Any questions so far? All right, so the next exercise asks you what would you expect to see in the ERCCs versus counts plot if you were examining a data set containing cells of different sizes. So this is another problem with single cell data analysis. The amount of ERCCs actually depends on the cell size. So in the normal experiment, if you're assuming that the cells are all of the same size, and then you can compare ERCCs between those cells. So if you have cells which are of different size, you expect to have different ERCC content in those cells. So it's important to account for this if you know in advance that you will have cells of different sizes.
Is there any guideline or criteria for proper amount of ERCC for cells?
Okay, so the question is, what's the amount of RCCs we should add to the cells in the experiment? I'm not sure about that. Tallulah, can you comment on this, on the amount of RCCs that should be used?
Essentially, if you have lots of money for sequencing, then ideally about 50% of your reads should be going to ERCCs, and then you'll get very good detection of your ERCCs as well as your endogenous genes, and you can use them quite well for normalization and stuff. If you can't afford that much sequencing, it's better to use a relatively small amount, and then you'll only get sort of about 10 ERCCs being detected across your cells, which isn't very many, but it's enough to do the sort of quality control stuff. But it depends on how big your cells are, what the absolute amount that corresponds to.
Is there any kind of reference data?
What's the question, sorry?
Is there any reference data we can get the proper amount of ERCC for specific cells?
Okay, the question is there any reference publication or data which you can use to define the amount of ERCCs you want to add to your cells? Tallulah, do you know of any?
In a word, no. I think typically people will run a pilot with potentially trying different ratios of ERCCs to endogenous RNA to decide to get to a point where they're happy with the ratio that they're getting. So to reiterate, Tallulah's point about this distinction between using ERCCs for normalization versus using them for QC, you would have quite different targets in mind. So I've recently seen data with using 1 to 2.5 million and 1 to 5 million dilution ratio of ERCC RNA to endogenous RNA, and that gives typically less than 1% of the reads per cell going to ERCCs. So that is useful for QC, but definitely not enough to do any normalization based on those ERCCs. You would obviously need to increase that ratio a lot from that point, so 1 to 2.5 million, to get to anywhere close to 50% or whatever percent of your reads going to the ERCCs.
And I think we should add here that the way the ERCCs are constructed is that you basically, so I think there are 96 of them, and they are constructed in a ladder, a concentration ladder, sort of spanning about 6 orders of magnitude. So that's why if you only have a sort of very dilute or few number of reads going into your ERCCs, you'll only get the few ones which are at the very top of the ladders, which have the highest concentrations. So that's why you need to have a lot of sequencing reads going onto the ERCCs in order to be able to capture the more lowly expressed ones, which is sort of what you need to be able to do proper normalization.
All right, so...
We have another question.
Oh, sorry. Where is the question?
Just to ask, how deep do you actually sequence per cell?
So the question is, how deep do we sequence per cell?
So yeah, so there is it really depends on the biological question. There was recently a paper from Sarah Teichmann's group where they showed that for across a lot of different protocols, at about 1 million reads per cell, that is sort of, after that you no longer detect additional genes. Right? But if you're interested in sort of having accurately measuring the level of expression in different genes, then you'll obviously need more sequencing depth. But if you're more interested in sort of identifying different cells, then most of the information is actually in sort of whether or not a gene is present or not, and then you can get away with much lower sequencing depth. So obviously, one of the most important trade-offs for your experimental design is number of cells versus sequencing depth. And exactly which point to land at there really depends on sort of what is the most important factor for your scientific question.
We have too many opinions. One popular experimental design at the moment, for example, is to use one 384 well plate and use one flow cell of high-seq sequencing on that. And if things basically go well, then almost all of your cells will get somewhere between 100,000 and 1 million reads. And that's, that's a lot of good information for many of the tasks that we'll want to do with it. Although for specific questions, as Martin rightly says, you want to tweak that one or another.
Any more questions? Okay, so we'll move on to the next chapter, which is cell filtering. So we have created all these cell filters by running the previous code chunks. So here we can apply those filters, and these are the names of the filters that we've created. And basically, in total, those manual filters that we used removed 210 cells from our data set. So it's actually quite a large fraction of cells, which we don't want to use for the downstream analysis. So we also want to mention that Skater has some default filters that you can actually, that are calculated when you run the calculate QC method. So you don't need to do anything to compute those values. You can use them straight away. And basically, these filters are defined based on these criteria. So you calculate the median absolute difference between the cells using the number of reads in each cell. And then Skater removes all cells where this distance is more than 5 times median of D. So also Skater removes all cells that are more than 80% of counts assigned to control genes. Is it still the case?
It doesn't remove them, but it does flag them.
Yeah, so it's still 80%, right?
That's the default, yeah.
Yeah. So if we use the Skater default filter, so actually to run this, we didn't need to do anything before. We can just run it straight away after creating the Skater object. And this filter actually removes only 6 cells, which is, again, sometimes this filter can work well. In our case, probably it's better to use our manual filter because it removed more cells. Also, Skater contains an automatic filter which is based on the PCA plot, but this PCA plot is based not on the expression values of your genes, but based on these 6 features. So these features, again, are calculated in Skater automatically. And then you basically, for each cell, you have these 6 features, and you can construct a PCA plot based on these features. So if we do this, this is the kind of plot that we see. And again, because the features are selected in a way that they take into account most of the information from your data set, again, you can see, and also the controls are included in these features. You can actually see, again, this batch, which was an outlier. And Skater has also a default algorithm which can identify the outlier cells from this plot. So you just run it, and it will tell you that, basically it tells us this orange cells are outliers, and the rest are good cells. So this filter removed 73 cells from our data set. And now we can actually compare these different filterings with each other. So for this, we use a lemma package which contains the Venn diagram function. So Venn diagram is usually when you have several sets which can overlap, and you want to basically see what is the overlap between each pair of the sets. So here we have our manual filter, the default Skater filter, and automatic Skater filter. And what we see is that our manual filter basically picked up all the cells that were picked up by the Skater default filters. Unfortunately, those filters do not overlap. But again, it's always a good idea to look at your data with your eyes and basically finding out bad cells by yourself. And you can see that our filter is much stronger in this case. Any questions? So once we've done the cell filtering, now we can do the gene filtering as well, because some of the genes are completely non-informatives. They might contain a lot of zeros. So for this, we will first use this plot QC function, which plots the top 50 genes with the highest expression in your data set, just to have a rough idea of how these genes look like. And from here, we can see that for each gene, we actually plot the number of total features in each cell, right? So here, for each gene, we have a distribution, and this distribution is a number of total features per cell. So what we can see is that these distributions are quite flat. That's what we say here.
Is this after normalization?
This is, that's a good question. Let me see. No, I think this is not after the filtering. This is all, we use all the cells here again, so we didn't filter it yet. But still, I mean, the main point is that we can see a few spike-ins here, which suggests that the greater dilution of spike-ins may be preferable if the experiment had to be repeated. So ideally, you would like to see endogenous genes in this list. But again, the distributions are not, they're quite flat, so there are not a lot of outliers. So basically, it tells you that the data is all right, the genes, the top 50 genes. And now we can do the filtering of genes which are considered undetectable. So for this data set, we define a gene as detectable if at least 2 cells contain more than 1 transcript from the gene. So if you have 1 cell with 1 transcript out of 800 cells, which means that this gene is actually not informative at all. You don't need to keep it in your data set. So that's the gene filter that we apply. And if we look at the number of genes that it can remove, this filter removes about 4,500 genes, which again shows you how sparse your matrix is. So a lot of the values are zeros in this kind of data sets. That's why gene filtering is quite important, because it makes your life easier for the downstream analysis, so that you have less dimensions in your data set. And basically that's it. So now we can, with this, where was it? So so we don't save the filtered data set, but we just save the same data set that we use throughout these exercises. But again, we have all the information about the filters in this use variable in this data set. So when we apply our manual cell filter and our gene filter, we have this number of genes and this number of cells. So what we need to do is just to save this object, and then we can use these filters whenever we want to do any downstream analysis of this data set. So that's, again, advantages of Skater when you have only one object and everything is contained in this object. So the good thing about this data set, it's not only contains UMIs, but it also contains the reads. So now in the next chapter, we would like you to go through the same steps that we did for the UMIs, but using the reads instead. So the read file is in this folder on your computers, and basically go through every step yourself and try to see whether the read quality control is very different from the UMI's quality control, and what are the differences between them. And if you have any questions, please ask. Again, you can either use the website, or you can use the RMD file, or you can do it yourself, whatever you prefer.